So, my empirical study looked at meadow and prairie voles and a possible reason for the differences in mating patterns which they have. The prairie vole naturally has a more monogamous relationship, uh, while the meadow vole tends to be a lot more polygamous. The experimenters examined the differences in oxytocin receptors in the brain, testing to see if the density impacted the style of mating. The gene which codes for oxytocin receptors was spliced in order to create a plasmid that could be cloned and then used for the experiment. The gene was surgically injected into the vole's brains. A sham group was used in this experiment and underwent the same procedure, undergoing anesthesia, being operated on, and then sutured. The voles were housed until 2 months after their surgeries had occurred. Then the testing began. Females were placed in a cage with a sexually experienced male for 6 hours, recording any mating behaviors that may have occurred. Following this, the females were taken away for 14 hours. Once this time period had elapsed, they were placed into a partner preference test, putting the known male tethered on one side of the cage and a stranger tethered to the other to see which one the female would spend more time around. The oxytocin receptor enhanced prey group mated much sooner than the control and the sham did. However, the frequency of mating did not increase. The meadow voles were vaguely affected, showing little to no differences in comparison with their control and sham groups. The scientists think that the lack of results seen with the meadow voles could be a cause of the oxytocin affecting many regions of the brain, not just the part which was injected and therefore affected. It was suggested in the study that in future studies, scientists should increase the amount of sites which are used for injections to other parts of the brain, just to see if any other parts may affect the style of mating.